Welcome, everybody. Welcome.
I was on one of like the really fast.
Jeremy bought one for himself.
We are here for a very happy occasion. The exit seminar for Darien Satterfield. No. I want to tell you just a couple of things about Darien. First, I want to tell you about the first sort of thing that I ever learned about her. She wrote me when she was a master's student at Cal State Long Beach. She told me about this experiment she did for her master's degree. It involved going out in the kelp reefs, catching surf perch nearby where she was there in Long Beach and also over at Catalina, bringing pregnant female surf perch. It's like the only live bearing teleos fish on the planet, pretty much. Back to the lab where the females had babies. She then raised these babies in labs and behaviorally scored them in various ways for their sort of skittishness in response to predators. Did this absolutely, you can't imagine how hard it would have been to do that master's work, to do that project. It involved going out there, rain or shine, in Southern California, it never rains. But going out there in shine or overcast, catching these fish, which are never easy to catch, bringing them back to the lab, taking care of them well enough so that they have their babies, raising the babies, making them happy, doing these experiments, getting great results, writing a paper and publishing. But man, this is my kind of scientist. And that really is the way she is. She's just sort of, whatever it takes to get the thing done, she sort of gets really good at it. She really has this amazing capacity to learn things and get better and better and better at them. Darien is also a member of a certain generation of students. We heard about one of those last week. Students who were sort of in key points in their career as a graduate student when that freaking COVID pandemic happened. And the other thing I want you to know about Darien is that she is just probably the single most resilient student I've ever had. Just no matter what happened, she came back with a big smile on her face the next day and retooled this, retooled that. Who needs that project? I'll go do this project. I mean, we had more fun doing those kinds of exercises. But really, it was just one thing after another, her showing this time and that time, showing that she could learn how to do this and even just taking on whole new sort of skill sets that hadn't been expected. Just really an amazing experience for me. It's been wonderful. And in the last year, she's done just an amazing job in particular. When the dust finally cleared on the pandemic and everything, God, look out. She just took over and it's just been great. So I'm thrilled to have you give this seminar. Take it away.
Thank you, Peter, so much for that introduction, and thank you all for being here. It's so good to look out and see so many faces that I care so much about, and I'm so thankful for the time that I've gotten with all of you. But today we're going to talk about fish, and I want to go back to the very start of my journey with fish. My very first day in ichthyology, the professor comes into the room and he says, "Tell me what makes a fish a fish." And we start answering things like fish have gills, fish have fins, fish live in the water, they have scales. And all of these rules have exceptions, right? Because there is no one way to be a fish. Fish are incredibly diverse. And this has been the motivation for a lot of the work that I've done throughout my time as a grad student. Because surely there must be a limit to just how weird a fish can be, right? How do we get to this place where you have fish with jaws that are almost the whole length of their body or spines all over their body? Fish are just weird. And I've always been interested in how that weirdness happens. Luckily, I'm not the only one who's interested in that, though. There's a group at PBS and the Shed Aquarium who have built a simulation that you too could go home and play called How to Build a Fish. And in this simulation, you can choose the body of your fish, and the options you have are a rocket body, and those rocket bodies are super great for cruising around the upper reef. Or you could choose a pancake body, which is really maneuverable. Those are great for getting in and around those coral structures. You could also pick a noodle body, and those are meant for hanging out in caves and ambushing prey, right? So you choose your body. Then you can choose your mouth. You can be a fish that gulps other fish. You can be a reef grazer, so you're doing a lot of biting. You can be a fish that bites other fish, or you can be a bottom sifter, a bottom feeder. And so you choose your head, you choose your body, and it spits you out on the reef. And on the reef, you then have to swim around and eat as many other fish as you can. So that's the goal. I want to see your scores later. However, if you have chosen a body and a head that do not work well together, you will get game over. Your fish has starved. Yeah. So in this simulation, I picked a rocket body for cruising, but I picked a bottom feeding head, and it just doesn't work well together. I wasn't able to eat enough fish to survive. But in the next simulation, I picked that noodle body, which hides out in caves, and I picked a biting mouth, and I did okay on the simulation. Because when different regions of the body work well together, we call that functional integration. And that is the basis of how to make a functioning fish, right? You need to have functional cooperation between the different regions of the body towards functions like locomotion or feeding. And sometimes when you have functional integration, that can lead to evolutionary phenotypic integration, or when traits in different regions of the body are evolving together through deep evolutionary time. They're evolutionarily correlated. Now, it's important to note that functional integration can lead to evolutionary phenotypic integration, but so can shared developmental origins or genetic origins can also contribute to evolutionary integration. Okay, but what does integration look like? Well, we're going to take a look at the pectoral fin and caudal fins of fish. And you're going to see caudal fin shape on the x-axis here and pectoral fin shape on the y-axis. And if they covary, we would expect this axis of covariation between these shapes. Where right in the middle of this axis, we have our sort of most average fin shape, our most average pectoral fin shape and most average caudal fin shape. And at either end of these axes, we have sort of the extreme conditions. Where at this lower extreme, you have really rounded fin shapes, really rounded caudal fins, really rounded pectoral fins. And at the upper extreme, you have more angled phenotypes, really pointed wing-like pectoral fins and really forked caudal fins. And in this case, it turns out that this lower extreme is really great for maneuverability. They're high surface area, they can experience a lot of drag, and that helps with turning. And that upper extreme is really great for endurance swimming because they experience less drag, so they're not as energetically costly. And so in this case, this axis of covariation happens to relate to this functional trade-off, but that doesn't always have to be the case. When we talk about integration, we just mean the most dominant axis of covariation between these traits. Okay. But if we think about this as a morphospace, where right in the center we have our average shapes, what becomes apparent is that that axis of covariation is a pipeline to outer morphospace. It can lead us right to the most extreme conditions. And the way that that happens is in an integrated system, when one trait in that system experiences a direct source of selection, any traits that are integrated with it will be pulled into that novel region of morphospace as well. The converse of this, though, is that when a trait in an integrated system experiences stabilizing selection, the whole system can be constrained in its diversification. But you might also notice that there are off-axis regions of morphospace that are of outer morphospace, where you have extreme conditions, but they're not along that axis of covariation. So how do we get here? Well, the only way to get here is that all of the traits in the system are experiencing their own direct sources of selection, and they're evolving independently of one another. So that is the way that we get to these off-axis regions of extreme morphospace. So we have two ways to become morphologically weird, right? We can follow these axes of integration to outer morphospace, or we can evolve our traits independently and get to outer morphospace that way. Now, there is a body of literature, and it's a growing body of literature, that talks about these different evolutionary pathways and their contribution to morphological diversification. And some of these papers say that integration is a really dominant pipeline to producing some of the most morphologically diverse systems that we can find in nature. Other papers say that modular evolution or the independent evolution of traits is the most important factor in organisms being able to diversify. But the most recent body of literature says that it's actually a combination of both of these two pathways working together to lead to the most diverse systems like reef fish. And so in my question, we're going to be asking that that is the question. What are the relative contributions of integration versus independent trait evolution to morphological diversification in reef fish? So in contemplating this question, Peter and I had an idea. And this idea starts with the goal of characterizing evolutionary pathways to morphological novelty. If we can characterize the pathway that fish take to become morphologically novel, we can determine which of these pathways happens more often, right? Okay, so the foundation of this idea is that if you know a species' position in morphospace, you can do two things. You can calculate its distance from the average shape, the average condition, and we can consider that morphological novelty. For species that are further away from the mean shape, we're going to call those more morphologically novel. And species that are really close to the average, we're going to call those less morphologically novel. Also, if there's an axis of covariation between traits, we can calculate residuals from that axis. And the closer you are to that axis, the more evolutionarily on trend you are. And the higher residuals you have, the more off axis or evolutionarily mismatched you are. So for example, a species that falls here is really extreme morphologically. It's really far away from the average, but it has really small residuals. So we would say that it reached that morphologically extreme condition along that axis of integration. Here's another example of a species that is also quite far from the morphological average, but it has really high residuals. So we would say that it reached this condition by evolving its traits independently. Okay, now here's where the fun part comes in. If you have these two traits, you can plot them against each other. And now you have a space that you can divide into different areas and start to identify where species are falling. So let's imagine we have a morphospace and some species are going to be right at the average, right? So they're going to be 0 for distance away from the centroid of that morphospace. Other fish are going to be really far away from the distance of that morphospace. If we divide that from 0 to the furthest away species in half, that's where this is going to be, right at the half range of all of the possibilities. Okay. And so in this lower right-hand quadrant, we have species that are very close to the average morphologically, and they have really low residuals. So those are our species that are morphologically average, and they tend to be abiding by those axes of integration between traits. You can also have species that are morphologically average, but their traits are having a tendency to evolve independently. So they're leaning towards those traits being morphologically mismatched from each other. You can have species that are morphologically novel, that are very far away from the centroid, that are evolving along those axes, those integrated axes. And you can have species, and you can have species that are morphologically novel, but they have very mismatched traits. Okay, so now we can start to identify which forms of these morphological novelties come up more often. Okay, but remember we talked about fish are weird, right? Fish are super diverse. And it's actually going to take a lot more than just two traits, like the pectoral fin and the caudal fin, to describe morphological diversity in fish. And so we start to realize that answering this question about which of these evolutionary pathways contributes to morphological novelty, that's actually a much bigger monster than we had originally intended. But that's okay. We're going to just keep swimming, and we're going to break this question down into its parts, starting with morphological diversification in reef fish. So we're just going to start just addressing that problem by itself. How do we describe the morphological diversity in reef fish? And then, once we've done that, we can tackle whether or not there are axes of integration or covariation between these morphological modules. And once we establish that, we can compare how closely species fall to those axes of integration versus having high residuals and a lot of trait mismatch. Okay, so let's start with this first aim, describing the morphological diversification of reef fish. Okay, so let's think about what we know about a fish and all of the ways that fish can vary. And one thing that we talk a whole lot about in Peter's lab is the jaws. And in the jaws, we know that there's this axis of fish that do a lot of biting. And so their jaws are kind of made for crushing corals or urchins, hard things, right? And then at the other end of the axis, we know that there are species that do a lot of suction feeding. And so their jaws aren't particularly strong, but they're big and they can pull in a lot of water and overtake their prey that way. So we know that the jaws are an axis of morphological diversity in reef fish. We also know that there's a ton of body shape diversity. Reef fish can be very laterally compressed and deep bodied like a CD, or they can be elongate like a torpedo, like that barracuda is. So we know that body shape's a good trait to look at here. And then there's also the fins, and this has been one of my favorite things to think about during my PhD. And the fins can be incredibly diverse. You can have caudal fins that are really rounded, high surface area. You can have fins that are really forked. The dorsal fin gets really crazy. It can be split into two fins. Some don't even have spines at all. Fins are wild and way underrated. We should think about fins more. So we have these different regions of the body that we can look into to explain morphological diversity in fish. But the first step of doing this was to prepare a collection of specimens that spans ecological diversity in reef fish so that we can capture all of that morphological diversity. And what we did was we assembled a collection of 93 species from 37 families, and this is over half of all of the reef fish families. And we specifically chose species that span the trophic diversity in fish, habitat diversity in reef fish. This is a really ecologically diverse sampling of reef fish. In total, we have 294 specimens. On those fresh specimens, we took some measurements like jaw protrusion. We did that before we fixed the fish. And then we also dissected out the muscle that's responsible for some of that biting power in reef fish, the adductor mandibulae. But after we took those fresh measurements, we cleared and stained them. We fixed them and cleared and stained them and took lateral images of the body shape of these fish. And so you can start to see some of that body shape diversity that we have in these fish. Now, in addition to the body shape, we also zoomed in on the jaws, and we got a close-up picture of the jaw anatomy of the fish. We also pinned out the pectoral, the caudal, the dorsal, and the anal fin, and took pictures of those fins as well. So now we have this image collection that describes these morphological, or that we can use to describe these morphological diversity in reef fish. The question then becomes, how do we measure morphological diversity from these images? So let's talk about the jaws first. In the jaws, we took 12 linear measurements in addition to those fresh specimen measurements that we took. And most of these are things like lengths of the dentary, of the maxilla, premaxilla. But we also created a coordinate system here where we have a vertical line through the center of the eye and a horizontal line down the midline of the body. And that allows us to determine where positionings of different joints are. So this is the lower jaw joint and the joint between the palatine and the maxilla. And we can determine whether that joint falls forward of the eye, behind the eye, in front of the eye. So we can start to see how the jaws are positioned relative to other features in the skull. Okay, for the body shape, we placed 9 fixed homologous landmarks around the body. And these landmarks allow us to approximate the body shape of the fish. So throughout the talk, you're going to see these sort of odd looking gray shapes. What those shapes are is that center of the body of the fish. Okay, remember I went on for a bit about how weird fins are? It creates a problem for how we measure them. With fins, you can't often find homologous landmarks in all of these, with all of this diversity in reef fish. So what we decided to do here was use semi-landmark curves around the area of the fin. So we have the jaws, the fins, the body, but they're all measured in slightly different ways, and that's something we're going to have to account for here in a little bit. Okay, so let's now tackle this first aim. Now that we have these measurements, we can talk about the diversity that exists in these modules. Starting with the jaws, we took the measurements and we supplied them to a PCA. So what you're going to see here is the first principal components, or the first axis of diversity, and then the second axis of diversity in that module. And here's what the jaws look like. You'll see that that first axis of diversity in jaws represents how small and strong and robust those jaws are. Usually they're more forward as well, to jaws that are very long and thin, but big jaws. So we have small jaw extremes and very large jaw extremes, large thin bones, very long too. The second axis of diversity that we capture is where those jaws are positioned. So you'll notice at the lower end of PC2, we have jaws that are in line with the eye, forwarded and in line with the eye. And as you move up, those jaws drop underneath the eye, which you can imagine could be really important for different diet categories, right? Like if you're a planktivore and you really need to be able to see those little things in the water, it might make sense to get that jaw out of the way. Okay, Body shape diversity is also pretty interesting. So again, you're seeing the shapes that kind of map to that center region of the body. The largest axis of diversity that we find here is just whether or not you are short and deep bodied to long and very elongate thin bodied. So that's your primary axis of diversity. But the other thing that we capture here on the second PC is how tapered you are. So some fish have this body shape where the body tapers quite a bit towards the caudal. And the other extreme of that is that fish are very boxy. So they're kind of square all the way back to that caudal fin. I won't take you through each of the fins individually for time's sake, but I do want to talk about some of the big axes of diversity in these fins. Pectoral fins can either be very rounded or very wing-like and angular. Caudal fins are either very rounded or very forked. The dorsal fin, I think, are super interesting. Most fish occupy this kind of small region where you have low-lying, sort of elongate, continuous dorsal fins. And the way to get a weird dorsal fin is either to split your fins. Some of them have really extreme conditions like a single spine and triggerfish, or you can lack spines at all and just have a really short, kind of rounded area of the dorsal fin. And then the anal fin, the primary axis of diversity is continuous elongate fins versus very short and almost triangular fins. Okay, so we have lots of diversity in reef fish morphology. And then the next question becomes, in that diversity, are there axes of covariation among these modules? Can we identify integration among reef fish morphological modules? Okay, so remember that the way that we want to do this is by taking these morphological modules and asking if there are axes of covariation between them. And we're going to phylogenetically correct this to account for the fact that a lot of species are very closely phylogenetically related. So we're going to use phylogenetic integration as our measure of axes of covariation between these traits. Okay, we have 6 morphological modules. That means there are 15 combinations of traits. And so I'm going to show you the correlation coefficients for these 15 combinations. And I don't want you to worry too much about the numbers. It's going to be a lot of numbers up there. What I want you to notice is how many of them are going to be in bold, which means they are significant axes of phenotypic integration. Okay, so out of the 15, 10 of them are significant. So we have 10 strong axes, or we have 10 axes of covariation in the morphological modules of reef fish. And some of them are stronger than others. For example, one of our lowest is between the pectoral fin and the anal fin at 0.33. And then we have a strong one between the body and the anal fin at 0.77. But the average covariation, or the average phenotypic integration here, is 0.505. So I'm going to show you just a few so you can kind of visualize what these look like. This is the one that we've been talking about, the pectoral fin and the caudal fin. And you can see that typically more rounded fins do occur together, and more wing-like pectoral fins generally occur with more forked caudal fins. But there's a ton of variation around this axis of integration. So the integration is present, but there's evidence for some independent evolution around this axis as well. Here's another one that I find really interesting. This is between the body on the x-axis and the jaws on the y-axis. And typically, really elongate bodied fish also have really elongate, long, thin jaws. Deep bodied fish typically have very robust and short jaws, very strong jaws. And that's something that I want you to remember because it's going to come up again later. Deep bodied fish typically have very short, robust jaws. Okay, so in addressing this aim 2, do modules in reef fish make up an integrated system? We know that there are many significant axes of covariation in reef fish, and we know that there's lots of variation around these axes of integration. Okay, so now we can get to this aim 3. Does evolution of long axes of integration versus independent trait evolution contribute more to this morphological diversity in reef fish? And remember, the way we're going to do this is by mapping species onto this plot of morphological novelty versus that evolutionary tendency, or their tendency to hug those lines of integration. But we need one variable. We need one variable that captures morphological novelty in reef fish. But we have 6 morphological modules. So again, we're at this beast of a problem, right? we need to compress the diversity of these 6 morphological modules into one variable that represents morphological novelty. Okay, these PCAs are just axes of diversity. So you have a primary axis of diversity and a secondary axis of diversity. And it turns out that if you take those 2 and add 2 more, you end up capturing about 95% of all of the morphological variation within each module. So if you take those 4 axes of diversity and supply them to an additional PCA, you can generate an overall morphospace that accounts for that diversity in each of those modules. And so the loadings of this morphospace are the PCs from those individual morphospaces. And this is a little bit easier to think about if I show you where some of the fish are in this morphospace. So in this overall morphospace, kind of in this lower mid PC1, but low PC2 area, we have things that you think of as like typical reef fish, right? You have rabbitfish, butterfly fish, all of the things laterally compressed, deep bodied. What I think is really interesting is that as you go up in PC2, you can get more elongate at the low end of PC1, but we have this is the fish that I think is the weirdest in the whole data set, the frogfish, and it's really deep bodied, but it's not deep bodied in the way that these guys are, because these are laterally compressed. It's deep bodied and it's chunky, it's round, right? It also has really odd pectoral fins that are really underneath it, super weird fish. The other interesting thing that I see in this overall morphospace is that you have continuous dorsal fins in the middle and you get split dorsal fins towards either side, but they're not quite the same. You get a split dorsal fin with a really short spine section over here on a very laterally compressed deep-bodied fish. And on this end, you get a split dorsal fin, but they are both sections are more elongate and continuous. And you have a fish that's more elongate as well, and a little bit more rounded. So this morphospace separates fish that are similar to each other in body shape, but it also brings to light the way that having a split dorsal fin doesn't mean you're the same kind of fish, right? There are split dorsal fins on all kinds of different fish. Okay, so back to the goal. In this morphospace, we can calculate morphological novelty as the distance of each species from that average shape. Okay, so however far away you are from the average shape, that's going to be your morphological novelty. And so we did it. We took all of that diversity and all of those modules and compressed it down into that one variable that we can use for morphological novelty. Okay, but we also need one variable that captures the general evolutionary tendency, or how far off of those axes of integration you are typically falling. And so we're going to do something kind of similar. But because there are 15 possible axes of integration, we're going to calculate the residuals in each of those pairwise combinations. And we're going to take those residual variables and apply those to a PCA. And here's what that looks like. The loadings of this PCA are residuals from those pairwise combinations of traits. And so species that are way up here have really high residuals on this body and caudal fin integration. Species that are way down here have really negative residuals on that body and caudal integration. So it is the species in the middle that have residuals of 0, okay? So if you are a species in the middle, you are very on axis of all of these axes of integration. And as you get further out into this residual space, you are the species that are not abiding by those axes of integration. You're falling off trend of those. Okay. So we have the distance from the overall morphospace. We have the distance from the residual space. We can plot those together. And we have done it. We can take all of this morphological data and generate this biplot that's going to allow us to identify how morphologically novel a species is and the general evolutionary pattern. Okay, so let's do it. Let's ask how many species fall in this morphologically extreme area where traits are evolving independently. And let's ask how many species fall in this morphologically extreme area but they're along axes of integration. Also, we're going to ask how many species are just boring and normal, but what we really want to know about is how the weird fish get weird, right? Okay, so this is that same plot you've seen, morphological novelty, evolutionary tendency. Okay, let's start with this first quadrant. And remember, I told you that these are the half range, these divisions, we put them at the half range of morphological novelty. So between no morphological novelty and the most extreme fish, 66.7% fall in that lower morphological average area with pretty low residuals, okay? Which is not to say that there's not a lot of diversity here. They're morphologically average, but you have a ton of diversity within that region. You have, all of your kind of classic reef fish, angelfish, butterfly fish, rabbitfish. Also, I'm so sorry, wrasse people, but wrasses are just not that morphologically weird. They're just kind of normal. So we have a lot of diversity here, but they don't even make it past the halfway mark of the most morphologically weird fish. Okay, now we have our morphologically average species, so they're still not all that weird, but these are the ones that have a tendency towards evolving their traits independently from one another. And I'm going to talk to you about how a species gets here because it's really dependent on phylogeny here. Okay, that really most extreme high residual species, that's a planktivore, and it's Parantheus furcifer. And Parantheus is nested right within a clade of carnivores. And these are all in this morphologically average low residual space. But in a very short, on a very short branch length, Parantheus has been able to evolve this more forked caudal fin and a more wing-like pectoral fin. So relative to the things like its jaws and its body shape and its dorsal shape and its anal fin shape, the caudal fin and the pectoral fin are kind of weird. So it's been able to evolve those two traits independently of some of those other body morphologies. So that's why it's up there. Okay, so these are our morphologically not very weird fish. So now let's get into the cool guys, the morphologically novel species. And we're going to start up here. Morphological novelty through trait mismatch or kind of independent evolution. There are 8.6 of the species in the study that fall in this range. And I'll show you who some of them are. We have things like cowfish, which are really weird, very square body. Remember we talked about the extremes of body shape? This is that kind of square extreme of body shape, but really rounded fins. Lionfish, which has a kind of typical grouper-like body and mouth, but the fins are wild, right? The fish that I think actually characterizes this area quite nicely doesn't look all that weird when you first look at it, but when you start to think about how the traits are going together, it's odd. Remember what we learned about deep-bodied fish? They have small, robust jaws, but not this guy. This guy has pretty large jaws relative to its body design. The dorsal and anal fin on a typical fish will be very kind of mirror images of each other. But on this one, we have this real short, weird triangular dorsal fin and this really like elongate anal fin. That's odd. Usually deep-bodied fish have rounded caudal fins. This guy's got a more forked caudal fin. Just like everything about this fish is just mismatched. And so that's why it ends up in that area. Okay, so we've got 8.6% of our species there. In our last area, morphologically extreme fish along axes of integration, we have 15.1% of the species that fall in this area. Here are some of them. Our Antinarius falls here, but it really hugs that line right there. It's that one right there that hugs the line. So I'm not going to put too much stake into that guy. We're going to talk about one that's more well within this region. This triggerfish is way more extreme than it looks. Like you look at a triggerfish and its trigger is the spine is obviously kind of a weird thing, but otherwise it looks kind of like a normal fish. But their jaws are some of the strongest jaws in reef fish you can find. Super, super robust. They're definitely at the short, robust jaw extreme. Also that jaw muscle, some of the largest jaw muscles of the reef fish family. Their fins are at the very rounded extreme. Their body is at the deep bodied extreme. So everything about this fish is really extreme, but it's along the axis of integration that relates to being a really strong biter and a really strong reef associated fish. So this fish has followed those axes of integration right into being a functionally really awesome biter, really awesome reef fish. Okay. So when we compare the different regions of this plot, we can see that most species are morphologically average and generally abide by those axes of integration. But if we compare our morphologically novel species, we can see that morphological novelties come about 1.7 times as often along axes of integration as they do through independent trait evolution. And so we have an answer to this question for reef fish. It turns out that morphological novelties are rare, but when they do occur, axes of integration usually lead to them about twice as often as independent evolution of those traits. And so how do fish get so weird? Well, probably integration is a big source of some of those morphologically weird conditions. But because integration is present, but not overwhelmingly strong, fish are still able to evolve traits independently. And so we can get cases where we get novelties that occur through independent trait evolution as well. Okay. So remember we talked about the average integration between these traits being at 0.505. Well, it turns out that that's not all that weird for vertebrates. For vertebrates, this is a study by Connor et al. In 2015, and for vertebrates, the average phenotypic covariation is right at 0.5, and it's a little bit lower for plants and a bit more for invertebrates. So for fish, for it to be 0.505 is real expected. And so we wanted to think about how much the strength of that integration, that number, is really contributing to the patterns that we're seeing in how fish become morphologically weird. And so just as a final step that we did, we simulated Brownian trait evolution using different covariance correlation matrices. One where the integration is 0, one where the integration is 0.5, one where the integration is 0.9, and then we did final simulations. We did final simulations using the reef fish integration structure that we observed in nature. And we ran each of these simulations 100 times. This is that same plot that you've been seeing, morphological novelty against the evolutionary trend. And what we find is that when integration is really strong, by design, you can't get into those off-axis regions. The integration is too strong. It holds those species to that axis of covariation, right? But you get more morphological novelty when integration is strong than you do in any other case. When integration is present but moderate, you get species that fall in both regions. You get some morphological novelty through trait mismatch, and you get some morphological novelty along those integrated axes. And when integration is absent, you get almost no morphologically novel species. You do get a few, but they tend to fall right along the edge of morphological novelty through trait mismatch. Okay, so novel morphologies occur more often when integration is stronger, but under moderate integration, this is what allows for species to become morphologically novel in two different ways: by having odd combinations of traits and by having highly integrated traits. Okay. So just to wrap this up, remember that in reef fish, we found that morphological novelty occurs 1.75 times as often along axes of integration as it does through trait mismatch. So in our final simulation that we did under the integration structure of reef fish, we calculated the same ratio for each of those 100 simulations. And what we find is that in nature, we see a lot more integration than the simulations suggest we should see. So in the simulations, we find that highly integrated species only occur 0.86 times as often as highly morphologically mismatched species do. And so in thinking about why we're seeing more highly integrated species in nature than these simulations suggest we should see, it might be because Brownian simulations don't assign adaptive values to these different phenotypic outcomes. And we know that to build a functioning fish, you have to have trait cooperation. And it could be that those highly mismatched phenotypes could theoretically occur, but they would not be very adaptive in nature. But those highly integrated phenotypes can produce a fish where everything about that fish is really designed to highly specific ecologies, like being a really strong biter on a reef. Okay, just to summarize everything that we've talked about today, in reef fishes, there are many axes of integration among morphological modules, and the integration is present, but it's moderate. It's not overwhelmingly strong integration. That moderate integration is what allows reef fish to achieve extreme phenotypes, both along integrated axes and through novel combinations of traits. In reef fishes, integration is the more common pathway to novel phenotypes, but in Brownian simulations, it suggested that odd combinations of traits should be more common than highly integrated traits. So it suggests that in nature, it's possible that those highly integrated traits are more adapted than highly modular traits. Really excited to go forward in my research career. I'm going to Florida Atlantic University, where I'm going to be doing a postdoc under two really awesome professors, Marianne Porter, who's a really cool mom in science, and I'm super excited to learn from her experiences. And then Ashkan Fahimpour is a really cool scientist that does a lot of machine learning tracking of fish in situ, which is really hard to do and really cool, and I'm excited to learn that. And with that, we're going to do questions. And if you want to stick around, I'm going to do acknowledgements after the questions, if that's cool. Yeah.
That was such a fun talk. I learned a lot about fish. Thank you.
Yeah.
I have, as I often do, a question about statistics. I was intrigued when you were putting together your different principal component vectors, because obviously the first PC explains a lot more variation in morphology than the fourth one. So how did you weight the different axes?
Yeah, so we are taking all of these axes and throwing them in another PCA. And all we're asking that next PCA to do is tell us which of these dominant axes from each of the 6 morphospaces is explaining most of the overall morphological variance. So I think part of what washes out is if you have an axis within a morphological module that really doesn't explain all that much variation, it's not going to come through as explaining very much variation in the overall PCA.
It makes sense. I wonder a little bit if you weighted them by, in some way, by their importance, you might get more, the simulations might do a better
Totally.
job.
Yeah.
Something to think about.
I think that's a really good idea. Yeah. Make more dominant axes contribute more to that overall morphospace. Yeah. Yeah.
Yeah, very cool. So in the analyses where you sort of split things in the 4 quadrants,
Yeah.
right?
Mhm.
Those midlines are essentially like they're halfway between the fish on each extreme, right?
Totally.
So it seems like where those lines are drawn depends on how extreme a fish you have.
Yeah, for sure.
So I just wondered if you thought about like deleting your most extreme fish.
Yeah.
Or extreme fishes, and how that changes, or does it change the conclusions that you draw about what fraction of them are in this quadrant versus this quadrant, and so on, or is it not making any difference?
It's so funny that you say that because I had not had that idea until about an hour before this talk, and I was like, oh, I can really do that. Yeah, I do. I think it's a great idea because I do. The most extreme species that you have defines where you draw the cutoffs. Those cutoffs are a little bit arbitrary. You could do it, and your interpretation of the results changes depending on how you do it. I have a version of this where I've plotted the axes at the mean morphological novelty and mean residual. But what you lose in that is how many species are really average in morphology. So it's a little arbitrary where you draw the lines, but I think it's kind of fun to play around with like the different conclusions that you can pull. But that's like next step for sure.
So if I can follow up,
Yeah.
I thought about the mean thing too and landed exactly where you did on that. But I guess I was thinking like, how do you pick the fish that you use here? And is there any reason, like, are you biased towards picking the weirdest fish you can find? Or is this some sort of other data set that's sort of like neutral to how weird fish are?
Yeah, so this data set actually originally came from, like, the jaws were the first step of this in a paper that we all as a lab worked on, and postdoc Mike Burns in our lab led. And we built this collection of reef fish around looking at diversity in the jaws. And so if anything, I think it might be biased towards capturing odd jaw morphologies. But body shape and fin shape came later. And actually what determined what went into this sampling of fish was fish that had good fins mostly. The jaw data set actually includes about 20 more species that you don't see here because their fins were too torn up to use. So everything that had a good good fin images from that jaw data set got applied to here. But yeah, I could see how like we wanted to capture the full spectrum of jaw diversity for sure. Yeah. Yeah. Yeah, Joe.
Wonderful job. Honored to have been Pop Bio mentee. I was curious, it seems like a big part of your project was you had those two PCs and you ran that regression, and then got residuals off of that.
Totally, yeah.
Right? I was curious for that axis, it was the covariance axis you called it,
Yep.
right? I was curious, have you played around or thought of like maybe nonlinear interactions where maybe on some points, like earlier on, maybe it's a little easier, a little bit of a step in one direction leads to a little bit easier of a step in another direction versus maybe farther down the axis?
Oh, I love that. I had not thought about that, but I do think that that's a really interesting idea. Yeah, because you can imagine like if you adopt a slightly weirder body shape, it might be the case that that helps you in changing your fins a bit,
Right.
right? Yeah.
Yeah.
So I can totally see that being something that comes into play here. Yeah, that's an awesome idea.
There's a question online.
Okay.
How do you expect your results to change with climate change? Do you expect particular morphologies to perform better under more extreme climate conditions or even to see more extremes in the future?
Yeah, that's a really interesting point. I think it could go either way. Like if you have something about your body design that's really temperature dependent, like I kind of feel like the jaws are going to be what's most affected by this because a lot of prey items like corals are really dependent on what's going on with temperature, right? And so I don't know, I feel like I kind of expect to see maybe a condensing of some of those more extreme morphological conditions. I imagine that if you're highly specialized and really extreme for biting corals and the coral dies because it gets too hot, that really specialized condition might not make it. Yeah, that would be my intuition on that. Yeah. Yeah, Megan.
This is related, but has there been any studies done on extreme morphologies related to species extinction?
Ooh. Oh, that's so cool. I don't know for sure, actually. I bet that there is. God, there's gotta be some. I don't know if it's in fish though. I kind of feel like maybe insects would be the place to go for that. Or like, yeah, maybe even mammals. But I feel like there's been a lot of like looking at morphological integration in mammals more so than like any other group. So if we're asking like specifically how highly integrated phenotypes are affected by climate change or like environmental shifts, I think mammals might be the place to go. There might be something. I haven't seen it though yet, but it's really cool. Yeah. I saw Jenny first and then Andy.
So I was wondering, given your results and findings, which this has been a super fun talk. Thanks.
Thanks.
If PBS was redesigning a 2.0 version of the game, what advice would you give? I mean,
Yeah. Okay, so think about that sweeper, right? It's that really kind of tapered bodied fish, big old eye, big jaw, deep bodied fish, big jaws. Obviously, weird combinations of traits do happen and they can sometimes work. And it would be kind of cool to be like, you picked the weird combinations of traits, but like it happened to work for you because you found a system, like maybe those weird combinations are really great at eating urchins or something, you know? It'd be kind of cool to like throw in a little bit of chaos in there, I think. Yeah, Andy.
Yeah, that was amazingly cool.
Thank you.
Yeah, I really enjoyed it a lot, and I feel almost trivial asking a kind of a statistical question as opposed to a lot of the other cool questions here, but I did have this thing of, is it possible to even have a large mismatch and yet a small distance from the mean?
Okay, so
Isn't statistically the case that if it's a large mismatch off of the line, it has to be a pretty large distance from the mean?
if you're not considering phylogeny, yes.
Oh.
But so this is,
Uh-huh.
I lied to you all a little bit. So it's easier to explain those residuals on a non-phylogenetic scale. But actually, when we're calculating those residuals, we're asking how much change has happened per branch length. And so you can't like that Parantheus, which is really high residuals,
Uh-huh.
overall, its caudal fin shape and pectoral shape are not all that weird to the average. But it's super weird relative to the things that it's phylogenetically closely related to. So like that's how that condition comes up.
Okay.
just hard to show visually.
Yeah, yeah.
So I have like a philosophical question, I guess, that I'm interested in your thoughts on. So in your kind of split your 4 quadrants, and you show that under Brownian motion, that integration is where you get most of that drive happening to you. Would you expect those the same quadrants to have the most species if it was under selection? Or how do you think selection would maybe shift which of those quadrants is getting,
More.
the most species?
Yeah, okay, 2 things. I still think integrated phenotypes are going to be selected for more often than morphologically mismatched phenotypes are. But the thing that I am not totally sure about is like, would there be selection against extreme phenotypes? Because being extreme is like a little bit of a risk. Or if being an extreme phenotype has allowed you to access some new part of your niche, could that be selected for? You know, like it could, and there could be both. Yeah. Yeah.
One more question online. You focused on reef fishes. I wonder how your results might be different for fishes in other habitats with different selection pressures, freshwater, open ocean.
Yeah. So Mike and I have actually been talking about this a little bit recently. I think that there might be more highly integrated phenotypes in freshwater systems, especially because things like everything about the body plan spins everything being highly adapted for migration. So things like reproductive mode in freshwater fish, I think could really be driving an evolutionary tendency towards some really highly integrated phenotypes. Yeah. Yeah.
You talk a bit about the phylogeny, so I'm curious about like how phylogenetically related all these species that you showed are. So is it, do you see one point of origin of like a weird fish and then all the weird fish coming from that, or like recurrent?
Yeah,
evolution?
I don't have like a specific number for like how many species are independent of each other in those morphologically novel regions. But if you look at those morphologically novel regions, it's pretty apparent that they're not super closely related. Like you have weird things like barracuda really close to that triggerfish in that one region. So I don't think that it's a single origin that's leading to these morphologically weird conditions. The one exception is that tetradontids, so triggerfish, pufferfish, that all of those are kind of weird. Yeah. Yeah.
Very cool, I saw that a lot about fish. I was very curious, so just combining a few questions was, in your simulation, say you had this red spread. Now suppose of that, for some reason, things on the 3 quadrants go extinct, so all you sample are from this 1 quadrant at the bottom left.
Okay.
And now you do the analysis again, you will still see species going into that quadrant because you sort of have this renormalization. I was curious if that's what you should be interpreting. Is novelty relative to what you observe? Like, I was just curious.
Yeah, so this is like novelty relative to, like, it's almost like a community novelty, and now like novelty relative to what already exists in the system. And like what happened, I think the only way to get at that would be what happens if you like selectively remove all of the novel species. Do you then see a large push to like repopulating that novel area, which could be super cool to do. Yeah, I'm not sure that I like totally have an answer for like how that would go, but I think that that would be the way to do it. Yeah. Jay.
Enough about fish.
Fish.
So I was intrigued by one of your setup slides where you compared the extent to which fish versus plants versus invertebrates were morphologically integrated. And actually somehow invertebrates were like ridiculously morphologically integrated, which doesn't feel like the direction I would have predicted.
Yeah.
And so I guess I'm wondering, and then plants are less morphologically integrated, which I feel like, you know, I don't know if I would predict that or if just plant traits are hard to measure or what.
Yeah.
But so I mean, do you think that those differences are real, or are they like people pick such different traits to measure that it's hard to really compare those? And if they're real, why are invertebrates so morphologically integrated compared to other things?
Yeah, I'll tell you the answer that the paper says. I don't know if I buy it though. The paper says that inverts are more integrated because their lifespans are shorter, and so they have like a shorter time span to achieve like the goal of reproduction. And so everything about that insect needs to be, I don't know, I wasn't, I
Is it
was,
only insects? Are those the only invertebrates
I don't
in the
know.
data set?
I
I'm
don't
just
know.
curious. Okay.
I don't, that's a good question. Yeah, I didn't find that answer super satisfying when I read that either. I like kind of think that segmentation in invertebrates might be like, I don't know, radial symmetry is one of the things that like, are you measuring integration between two arms of a starfish? Because yeah, you're gonna get like high integration. So I think, I don't know, I feel like symmetry in inverts could be driving this. Sorry, Jay, I'm not an invert person.
Tell me the paper so I can go read it.
Yeah,
Thank
sure. Yeah.
So I lost track of whether or not somewhere in there you even talked about how what we know about the developmental sequence and process might actually even have predicted something about which parts are going to be more integrated and
Yes.
which are more free to do something weird because they're the last step or something like that.
Yeah, so for example, the dorsal, caudal, and anal fin all originate from the same fin fold. And so because they have that like shared developmental origin, it takes more for them to evolve separately from one another. And like genetic pathways, shared genetic pathways can also be, you have to break those apart before you can evolve things independently. So there are, yeah, functional relationships, genetic relationships, and developmental, like shared developmental origin.
Because the anal fin was the one that was super correlated
I
to
know.
everything else.
Yeah.
And does that just make sense in some functional way or?
The anal fin is the weirdest thing to me
Yeah.
because it's like not a fin that you think of as
Yeah.
being used very strongly in like, you know, pectoral fin swimming or body caudal fin swimming. Like it's not a very like swimming, it's not the fin that you think about when you think about swimming. So I almost wonder if like the anal fin just kind of remains, okay. So fun fact, sometimes integration will break down when you have functional expansion. For example, when mammals develop flight, there is a like separation of integration between the forelimbs and the hind limbs. And so I almost wonder if like, because the anal fin is not doing the like important swimming things that some of the other fins are doing, it just kind of stays integrated. And some of the other fins, there's selection for the breakdown of that integration because of their like functional demands. Yeah.
Jeremy has a question.
If you could have the opportunity to go on a reef dive 50 million years from now, do you think that evolution would proceed and the average fish would still dominate and there would just be these weird fishes that occupy a few niches here and there? Or do you think diversity would continue and trend in a progression to where weird fish would become more normal?
It's a good question. I think the thing that comes to mind for me is like when you look at prehistoric fish, how like crazy they are. I don't know. I think it could go either way. I think you could like say you lose corals and fish have to figure out something new to do in order to like adapt that loss of that niche. You could get weird phenotypes from that, but you also could just get like a canalization of losing all of the extreme phenotypes. Yeah.
The
Okay.
question does assume that the Earth is still here in 50 million years.
Okay, so I think that we need to move on to acknowledgements for time's sake, but if you have other questions, I would love to answer them after the fact. Okay. So it has been an incredible, it's been an incredible 5 years in the program, 6 years since my interview. Thank you, Maddie, who took a lot of these pictures. These pictures are from our recruitment weekend hike every year, and I'm so sad that I'm not going to be at the next one. But I've had some really amazing experiences during my time as a grad student. I've gotten to learn how to be a better teacher, gotten to go to Australia, got to do field work in Bonaire, and I'm just so thankful for all of the amazing opportunities that I've had here as a student at Davis. And along with all of those opportunities, there are so many people to thank. So apologies in advance. This might be a little bit of a long acknowledgement section. Okay. I want to start with Peter, who has, I think, believed in me more than I believed in myself from day one. And like, as a first gen kid who like kind of like imposter syndromed her way through her master's program and then like, oh my gosh, I'm going to a UC, like we're gonna do a PhD. I feel like Peter had so much more belief in me than I ever had in myself, and I'm so thankful that he saw the vision. And just the most amazing mentorship, and Peter's ability to adapt to the needs of an individual student is something that I will take with me and carry on forever. I have the best lab. Oh God, all my lab buddies are just the most amazing people, the best friends and colleagues, and just, I love you guys. I think, I will never be more grateful for the lab that I ended up in. Yeah, amazing lab mates. And more than just being my lab mates, they're also just super good friends. We've had some really amazing time diving on the Great Barrier Reef, winning fish trivia. We've had some good times, and I really love my lab buddies. My cohort and Pop Bio broadly. Woo! I have the best cohort, the best group of friends. I'll never forget the very first day of the core. It was Graham's section. My car broke down like 2 miles away from campus, and I called Maddie and said, "Hey, can you come get me? I'm not going to make it to the core on the first day." And she did. And like that, I think just like I could call up any member of my cohort at any time for anything, and they would be there. And the rest of Pop Bio too, like I've had some awesome mentees, I've had some awesome mentors, and yeah, I will miss all of you so much. The professors and the staff, oh gosh, can we just talk about how great Sherry is though? Like, our whole lives would fall apart without Sherry. They're everything to us. All of the professors that I had in the core, my dissertation committee, Jay and Kate, Peter too, and my QE committee, just everybody who I've learned so much from and have had the opportunity to work with and get mentorship from. There's not a better group of faculty at other universities. I'm convinced of it. We are, as the Pop Bio grad group, we are incredibly lucky to have the mentorship that we have. Okay. Aww. There's one person in this room that I would not be here without, and that's my dad. Who, so he met me when I was 3, and he'll tell you that I was the most talkative kid on the planet, and with that came a billion questions every minute, and my mom was not going to be the one that was going to answer all of those billion questions. We'll get to her in a second. But he was, and the first question, science, I remember was we were listening to Smash Mouth's Might As Well Be Walking on the Sun. And I was like, Dad, how can people walk on the sun? And then we started talking about science, and every English test that I failed because it wasn't science, he helped me with. And people who believed in me, I think that there were times in my life when I really tried hard to derail my path, and he made sure I didn't. I'm very thankful for that. Love you. It's been me and my mom since day one. She was a teen mom, and I think there are so many women who are responsible for every single one of you being here today. And I think it's really a shame that there are not more young mothers in this room. I think it's a shame that us young women have to sacrifice our motherhood goals in order to complete grad school. And that's what she had to do to raise me, and she chose me. And I hope that y'all take that with you and do what you can to make grad school more accessible for young mothers. But I'll always do what I can to achieve as much because of what she gave up for me. She told me that if you're going to do science, you go all the way. And I always knew that meant this day, getting through this PhD program. Her love has always been unconditional. When I failed the trigonometry exam and at 20 years old and threw myself into her lap, she was there to hold and comfort. And I think that I couldn't have asked for a more supportive and better mom and family. My little sister, who is not here, but she is on Zoom. She was the first person that I ever got to practice playing teacher with. And I know that she hated that, but I really got a lot out of it. And I think, gosh, she's so sassy. If you think I'm sassy, you should meet her. And I think that her like spunk and attitude and just like encouragement through this has been a real game changer for me throughout this process.
What are you guys hugging there?
Well, that's
The dog.
The photo doesn't have his head in it. I hate this. It's our dog. He's crazy. He's a good dog though. I have so many other women in my life who are responsible for me being here today. My neighbor Lorraine, who was single-handedly the reason that I was able to have 2 dogs and still get through grad school. When I didn't have to leave campus to go home to let them out, it was because Lorraine did that for me. My best friend in the whole world, Lori, is my emotional rock. And Megan, who did all of my field work with me and just has been the most amazing friend. And just my mother-in-law, my grandma, my, Alexis and Catherine, who most of you know, I'm so fortunate to have the most amazing network of really incredible women that have supported me through grad school. Jeremy. I think one of the most career decisions that you can make is who your partner is. And I think being able to be successful in your career is 1,000 times easier with a partner that says, yeah, go do it, and doesn't hold you back and doesn't make you feel bad for having ambitions and goals. And that is the thing that I appreciate in Jeremy more than anything else. He has been my fieldwork engineer, my adventure buddy. He is just, yeah, the love and support that I have from Jeremy is definitely foundational to what I'm able to do as a grad student. And I'm so glad that we get to live together now. That's great. Last but certainly not least, there are so many little nieces and nephews. The babies in my life are where I found joy during grad school. Being their aunt is it's my biggest source of joy in this life. And I hope that I make them proud because they make all of me proud. And I know that some of them are going to grow up to be scientists someday because I can see it already. And I just, all of my babies, I love them so much. And thank you all for being here so much today. And yeah, if you have other questions, I'm happy to answer those.